Okay, so hi everybody. My name is Manja Alphonse. I'm a senior researcher at Wageningen University and Research in the Netherlands. And I'll be presenting about image analysis methods for plant phenotyping. You can see here in the circles the three focus areas of our university. That's food production, the environment, and the quality of life. And yeah, that's the mission statement of our university, to explore the potential of nature to improve the quality of life. And our university is focused on the life sciences, and we actually consist of two legal entities. That's the academic part, the degree granting university, and the research foundation where I'm employed. And in the research foundation, we do more applied research and also contract research. And I work in the group called Vision Plus Robotics. We are a group that's dedicated to computer vision, AI, and robotics, not just to agriculture, but also to other areas such as the environmental science, conservation, animal and marine science, and more recently, even sports. You can visit our site to get an idea of what we do. All right, so I'll follow roughly this outline. I'll first provide a motivation and then go over two practical use cases of phenotyping, which benefited from computer vision and AI. So what is phenotyping anyway? So it's defined as the quantitative measurement of the functional characteristics of any organism. We know that an organism, a plant or an animal or even us, that we are influenced by the interaction between the genotype and the environment. So the genes are what defines what makes us a variety, or what makes any living being what it is. And the phenotype is the functional result of both the genes as well as the environment in which the growth and development occurred. For example, if we go to a tomato plant, we can tell quite easily if it's a cherry tomato or a regular tomato based on the size of the fruit. So so we have here some of the phenotypic traits, like the fruit or leaf size and shape, then color, grouping or diffusion. And these traits vary as per the crop or the organism we are interested in. Why would we want to do this? So for a grower, it's important to predict the yield that they would have, and also for planning for the harvest. Whereas for the people who develop the varieties, it's also important to know which varieties make more sense economically. For these reasons, this is an important topic. And ultimately, everybody wants to maximize their profit. So in practice, the production systems are optimized for maximizing the output. So that's a point to keep in mind. Yeah. Now, the role of computer vision and AI here is logical, because compared to manual inspection or data collection, imaging is quick and non-destructive. And you can also get much more accurate analysis than by manual inspection. And yeah, deep learning has made it even possible to not define the actual features or criteria that you use for the evaluation, but simply provide data for training. And of course, compared to other means, image-based phenotyping can be used in very large cases, such as large greenhouses or large fields. So it can be used quite easily in high throughput cases. So we can first approach the problems. Of course, in the specific cases of a particular crop or plant, the information that we want to get out will be different. So we can bring in the domain knowledge. For example, in the case of tomatoes, we make assumptions such as the shape and color of the fruits, and also use other information like a ripe tomato is red, whereas one which is not ripe is green. And we can approach this situation by using all this knowledge and use, for example, color spaces in which the colors of the objects of interest are emphasized. This can work, we can see here by fitting circles, we can get quite a few of the tomatoes, but keep in mind that this method is sensitive to parameters and to the situation in the specific greenhouse. And so it may not generalize. And we can indeed see that there are false positives or some missed detections. And this was the reason our project called Rob2Pheno, or Robust Robotic Phenotyping, explored AI and deep learning for phenotyping in a practical tomato greenhouse. This was joint work with Ensasaden, which is a seed company, and it was co-financed by the foundation TKI, that is the Dutch government's funding agency for top sectors such as agriculture and medicine. So we see here this robot which would autonomously navigate through the greenhouse, and it's capable of autonomous operation, and it collects images from the set of 4 RealSense cameras. So we have the RGB and depth images, and here a couple of examples of the annotated ground truth. In this case, we want the foreground fruits, and for the single class, or with the 2 ripeness classes, whether it's ripe or unripe. And yeah, we need to keep in mind that in a greenhouse which is optimized for production, the tomato plants are moved along the growth process to be able to have as many as possible. So this makes our life a bit difficult. And the protocol used was that a group of plants of the same variety were delineated by these ribbons. But that makes it a bit easier because that helps in the image analysis. Yeah. The data set that we have is partially public. We have some annotated images which you can find from the link. Uh, and you need to keep in mind that each run of the Phenobot produces roughly 1 GB of images. So we have gigabytes and gigabytes of data, but the ones of interest are the images taken just before and after harvest. And this is what we will use to compare against our phenotypic data like the yield. Here you can appreciate from a sequence of images taken by the 4 cameras. So there's parallax and there's also repetitions. A particular tomato may appear in more than one image and at more than one time position. So we need to take into account all these factors before doing our image analysis. So our pipeline first used instant segmentation, mask our CNN for detecting the fruits. And yeah, we were also detecting the banners that delineate the varieties. After that, once we had both the RGB image as well as the mask which was detected by the deep learning, we performed a correlation-based registration method to be able to stitch these images together, or at least the detected masks, to be able to not count the same fruit more than once. And once we do this, we are in a position to compare the pre-harvest and post-harvest mosaics of a group of plants of the same variety, as we can see here. And we can see that the lower trusses, which correspond to the ripe fruits, are the ones which are in the pre-harvest but not in the post-harvest images. So we can base our calculations of the yield based on this group of trusses which don't appear in both grids. In terms of the results, we've obtained quite good results at the image level for the fruit detection, for the fruit as well as the ripeness. But the plot level yield prediction was not as good as we had hoped, but this was because we used an approximation in terms of the delineation of the varieties. If we were to be able to trace the stems and segment accordingly, then we could have. I expect that the correlation factor of the prediction of the yield would improve. Yeah, and we also had a prediction of the individual fruit weight. There were some of the fruits which were weighed individually, and this produced a relative error of roughly 13%. And yeah, these results are presented in these two articles and MSc thesis. Yeah, and this was joint work with these colleagues whose names appear here. Yeah, and related to tomatoes, but in a different project with a different partner. We also have the Plantalyzer robot, which is from the company Hortiki, which itself is a spin-off of the Dutch manufacturer Berg Hortimotive. And this is not a phenotyping robot per se, but it is for use by the growers. So this one helps to detect the fruits which are ripe, and also develop models for ripeness over time. Yeah, this one's already in the market, and it's being used. It's been purchased by growers in the Netherlands, France, and the USA. Okay, so I'll deviate a bit from the technical presentation. Um, this is a line that I really love because it is from the book The Little Prince by Antoine de Saint-Exupéry. It's one of my favorite books, and I highly recommend reading it. It was some time back included in the curriculum in high school level in many countries in Europe, and also in the 70s and 80s in the state of Maharashtra in India, but unfortunately no longer. Yeah, but this is a line that describes clearly why a flower is important to the breeders or to the people who develop this variety. It's because a lot of effort goes into creating a flower variety that the intellectual property of the breeder or the one that's developed this variety needs to be protected. And the registration typically requires expertise, which is very specific, that people have to receive samples of the flowers from the breeders, analyze them for novelty, make notes. But this can be, again, difficult if there's a very large volume being produced. So can AI predict traits that uniquely characterize a flower? And this was the rationale behind our project Modoma, or the morphological descriptions of ornamentals through machine learning that we worked on along with the two Dutch agencies, Floriculture and Nachttuinbouw, which are responsible for the registration of flower varieties here in the Netherlands. And this was also a public-private partnership, which was co-financed by the TKI Foundation. And what I found really exciting as a result from this project was that even though we worked with the data set, which was the records of these two companies, and it was not that we created an experimental setup based on use for deep learning, we found promising results and a promising role for AI. So we see here some examples of the rose and gerbera flowers from the two of them, from the two companies. And we can see that there's quite a lot of variation between the backgrounds, and in this case, from knockdown bow, we also have a reference with regards to the size, but not from Floriculture. So this was really it could be that somebody just takes photos of these flowers with his or her cell phone and keeps them as a record. And as per our findings, deep learning can still work. And here we have a list of the flower traits which were recorded by these two companies. So they are different from the two agencies, but also with respect to the flower species, there are quite a lot of traits about the colors of the flowers, because those are something that's quite important. But other factors like the pattern or the geometry of the petals. The pipeline that we followed first used pre-processing to get rid of the background and to also remove the leaves to be able to not confuse the models, especially the ones related to color. And we also had to discard those classes which were too small and where there were not enough samples for a balanced data set. And after that, we trained our classifier or regression models based on deep learning or machine learning. And we tried different architectures, but experimentally, we found that ResNet 18 was the best model. I will go a bit fast in because of time. Here we can see that for colors we can use the histograms of the colors as one of the features that describes the flower. And a deep learning classifier would typically be trained in this way by providing the trait label and the flower image as the input. For one of the traits, the color of the Jabra, we had this confusion matrix. I'm sorry, the label names are in Dutch because the original labels were in Dutch. But we had an accuracy of above 82%. And also for the rows, we had roughly 75% of accuracy. And in both these cases, what we can see is that most of the errors occur in cases like between orange and yellow, or pink and red colors, where there's always some kind of overlap, or not everybody may be able to tell the difference apart. And from the graph of the error rates, we can see that either deep learning or a traditional machine learning based on nearest neighbor or random forest classification, we can get accuracies of above 75% or sometimes even above 80%. And similarly, for non-color related traits such as the inflorescence, we can get really accurate results. Although, in this case, the data set's not balanced, but the performance was still good. Here's an example of a difficult trait, that is the diameter of the Jabra head. So in this case, there's there's some confusion between the medium and the medium large, but that's to be expected because again, while defining the data set, there needs to be a clear-cut criteria for the annotation. And again, for the accuracy plots for the non-color related traits, we had quite a lot of them with accuracy of 75% or above, and there were very few of the traits which were difficult. So yeah, what we can conclude is that deep learning worked well in the case of floricultural trait registration, even without a tailored data set. And yeah, both flavors of deep learning classification as well as object detection were found to be quite good in practice. And also that the data engineering, the pre and post processing based on the specifics of the use case can be quite critical to the workflow. And finally, it's a two-way street between the insights gained from computer vision AI and the domain knowledge, in that the actual results from AI can be informative to the core domain, but also the inputs from the core domain need to be taken into account for things like the experimental setup. Um, Before I conclude, I would like to mention that at Wageningen University and Research, we have a facility called NPAC, which is the Dutch Plant Eco-Phenotyping Center. And we have equipment for phenotyping at different levels, whether it's the open field or greenhouse or growth chambers. We have imaging equipment for that. So feel free to check this out and contact me if you have any questions. And yeah, we are also organizing a summer school in the end of June in Wageningen, for which registrations are open. So I would highly recommend you to register if you are close by. It is an in-person event. The QR code is here, but I can share the link in the chat. So that was all. Thank you for your attention. I'd be happy to take questions.